Hey, come run qPCR with me. Hey, let's go over some background first. Let's go over PCR, polymerase chain reaction. PCR is a technique used to amplify a specific segment of DNA. qPCR, quantitative PCR, or real-time PCR is basically the exact same process, just has a fluorescent dye that measures the amount. Now let's get started. Our first step would be to isolate RNA. Now, the isolating kit comes with multiple steps, such as the use of a lysine buffer and many washing steps. After isolating, we are going to read it in a nanodrop machine to obtain concentration and verify purity. We do a 2-steps PCR, which means that we first synthesize the DNA and then we do the qPCR analysis. Now, the synthesis of the cDNA, which is also known as complementary DNA, it's done basically by performing a reverse transcriptase reaction. And our last step is to do qPCR analysis. Now, we perform this in our lab with CyberGreen Master Mix. Now, let's talk a little bit about PCR in general. PCR, of course, is a process of amplifying a specific sequence of DNA. Now, it is done by 3 steps that occur in multiple cycles to amplify that segment. These steps include denaturing, which separates the strands, primer annealing, which happens when the primers attach to the complementary sequence on the DNA template, and then, of course, extension of these templates, which is done by polymerase. Now, in qPCR, this is done with a fluorescent reagent, which basically determines the actual amount of PCR products at a given cycle in real time. Let's analyze our data. Now, qPCR measures the increase in fluorescence as the reaction proceeds. Our threshold cycles, or our CT values, are the cycles where the accumulation of fluorescence signal became detectable. In this set of data, we had 2 samples. We're calling one treatment 1, and the other one was our control, which is DMSO for us. Now, the main question we're trying to answer here is to see if there is a full difference for an expression of a particular gene in 2 sets of samples. In our case, our target sample will be GLUT3. We need to normalize for any loading errors against a reference genes. In our case, it will be MAPK. For our analysis, we calculate the delta-delta CT method, which includes 3 steps. 1 will be obtaining the delta CT, 2 will be calculating the delta-delta CT, and 3 will be calculating the 2 to the negative of delta-delta CT. Calculating delta CT will be by adding the target gene CT minus the housekeeping gene reference. And we do this step for both of the samples. Our next step will be to calculate the delta-delta CT, which represents the change of expression of the genes of interest between conditions. Now, this will be done by obtaining the sample delta CT minus the control delta CT. To obtain the full difference of expression, we calculate 2 to the negative power of delta-delta CT. Any positive values will express a higher level of expression for that particular gene compared to the control, while any negative will represent any lower expression than the control. Thanks, that's about it. I hope you had as much fun as I did.